What's up, my people? Super quick cell organelles lecture video. Okay, first off, this little phrase here, the whole is greater than the sum of its parts. Okay, this is this crazy concept in biology that we like to say, hey, we've got all of these parts, okay, and they are compartmentalized and they can all function their own way. And when we put them together, they're this working whole. Now, the sum of all the parts is awesome, and we love that each of them have their own individual function. But we have a problem if one of those things is out of place. So, for instance, we have the mitochondria, you guys know, is the powerhouse of the cell. If the mitochondria doesn't work, we don't supply energy to the rest of our organelles, we drop down, okay? So this is this phenomenon referred to as emergent properties. And we like emergent properties because it helps us in evolution a little bit later when we talk about complexity, okay? The more you have things relying on other things, the more complex, the more diverse you can become. And that's just really cool as far as like biological systems are concerned. And the more diverse we get when we start to talk about environment, the more adaptable we are as an environment and as a population. Okay. So first off, I want to say that there are internal membranes that compartmentalize the cell. Okay, and what the point of those internal membranes are is to minimize competing interactions. So, for instance, you're not going to see a lysosome doing the job of a Golgi. That would be a competing interaction. We don't want them competing. We want them cooperating. Okay, so we want cooperativity as opposed to competition when it comes to a cell and its thing. Second thing that is really big deal is like this surface area. The surface area is actually where the magic happens, okay? So you'll see a lot of organelles that have these folds in their membranes. So their internal membranes are their own little folds, increasing the surface area, and so reactions are able to occur more readily in those locations, okay? So let's talk about a few of them, okay? We're going to hit these bad boys today. One, the nucleus. So a couple things to notice here is the nucleus, we're only in eukaryotic cells. When we talk about organelles, we're typically only in eukaryotic cells. So the nucleus is like the control part of the cell. That's because it has DNA. That DNA is going to dictate the shape of our proteins and the interactions of our proteins and all that stuff that would make up those organelles. Okay, we also have some that are involved in assembly and transport and storage. Those are the Endoplasmic Reticulum, Ribosomes, Golgi. And then storage is more vacuole, and then obviously lysosomes as well is like helping us like create broken down things and reuse those as nutrients or expel those as waste. Okay, so before we get going too far, here's our nucleus. Big things here is the shape of the nucleus contributes to its function. So we have its own little membrane. This is the nuclear envelope. And on the nuclear envelope, there are these pores located all throughout. And these pores are going to be able to allow the nucleic acid information in and out of the cell. Okay, so when we need RNA to leave the nucleus, it can squeeze through those little pores. But what we will notice about this is that DNA is too fat. It being double-stranded, it can't actually leave the nucleus, okay, and head to a ribosome, which is going to be what makes the proteins for us, okay? So here's our nucleus. He's awesome. We love him. He controls the cell, but really his DNA is really the driving factor of what things should be doing in the cell. All right, so here's our endomembrane system. We have the nuclear envelope. Again, that's a membrane holding our DNA. We have the endoplasmic reticulum, Golgi. A lysosome counts as an endomembrane system, and here's kind of what we'll talk about, is that we're going to use a lysosome to break things down, or we're going to use this endomembrane system to help us make a lysosome. Okay, a vacuole is going to be in that, involved in that, and that's typically only in plants. And then obviously the plasma membrane itself. And these things right here, so vacuole and plasma membrane, is kind of like where's our end goal? Do we need something to leave the plasma membrane? Do we need something to become a part of the plasma membrane? Do we need something to go into storage in a vacuole? And so here's one really big thing that we like to talk about. All of our cell organelles, they have to talk to each other. And so that means that we've got to ship things from organelle to organelle so that they can cooperatively interact. And so how do they do that? They do it via these things called vesicles. And vesicles have their own little lipid bilayer. And we'll learn about those in just a second and talk a little bit more about them. So here's some pathways that we can do. We got some information leaving the nucleus. It's going to go to the ER, probably the rough ER where the ribosomes are, and the ribosomes can make us proteins. Okay, we can go to the Golgi and we can package and modify those proteins the way that they're supposed to be. And then, we can send it as storage to a vacuole. It could be the end result be that we made a lysosome full of enzymatic digestive enzymes. It could be that, we actually don't need to make a protein, but we need information to get to our smooth ER to make lipids to help us build the plasma membrane, okay? So lots and lots of things going on here. And again, these are all relationships between parts of the endomembrane system. Okay, so let's talk about a lysosome. Big things with lysosomes, 3 big functions. 1, it can digest food for us. 2, it can degrade or digest dead organelles or organelles that don't work anymore. And then 2, it can just destroy an entire cell, and boom, you got a apoptosis where it's just programmed cell death and the cell just blows up. Okay, so here we go. A lysosome, it's got these hydrolytic enzymes in it. Okay, so hydrolytic enzymes, again, think about hydrolysis, hydro, water, lys, breakdown. So these hydrolytic enzymes are able to break down just a bunch of different macromolecules, okay, into their monomers. And so they are very diverse. They can break down proteins, fats, polysaccharides, nucleic acids, all that jazz, okay? And so there's 2 ways that it can break down stuff. It'll break down stuff as digestion. These are typically going to be the macromolecules that we're eating as food, the cell needs as food, okay? Or we can break down parts that are dead in the cell that we're going to recycle those parts and reuse them to make other organelles. So there's 2 big types here. Okay, we've got phagocytosis, okay, which is where we've taken in food from the cell and we're going to use a lysosome to break that food down. Okay, again, the food gets taken in and then it's inside of a vesicle. This is its own little membrane. Okay, we can also do this thing called autophagy. So phagocytosis, it comes in from the outside of the cell. Autophagy is it's an organelle that needs to be broken down. Okay, so those are the 2 types for a lysosome. Okay, the third type we said is this concept called apoptosis. And so with apoptosis, it's the lysosome is going to digest the entire cell. And then obviously the surrounding cells that are not being digested by their lysosomes, they can absorb those nutrients and help us with taking those nutrients away and not wasting anything. But an example here would be a tadpole has a tail. It's going to lose the function. It doesn't need a tail because it's going to grow legs eventually and become a frog. So we don't need that tail anymore. And so we will go through the process of apoptosis, and that will break down that cell for us and let it get reabsorbed back by the froggy. Another organelle that is super important that we like to talk about, the Golgi, okay? So the Golgi, again, it's an internal membrane. It's got its own little lipids, and then it's got its own little membrane. And again, surface area increases reaction. So we have lots of things going on that Golgi, okay? We have a Golgi that is responsible for modifying or packaging or making sure proteins are in the right shape. So that primary, secondary, tertiary, and then quaternary structure of a protein, the Golgi is responsible for that. It modifies the protein so that it is the correct shape. Okay, so here's how we have the Golgi. There are 2 sides of a Golgi, a cis face, which is where it receives the primary structure of a protein. We also have the trans face, which is responsible for shipping. So I like to think of it as cis as receiving, and then we've got trans, which is transporting the actual finished modified protein so that it's actually functioning. Now, how do we get information to the Golgi and ship information away from the Golgi, these proteins? Well, we put them in a vesicle, we transport them to the Golgi. When we're done with them, we put them in a vesicle and we ship them where they need to go. Okay? All right. Next, okay, we have ribosomes. Really simple thing about ribosomes for now, because we talk about it a lot later as well, is that they're responsible for building our primary structure of our proteins, okay? So just a strain of amino acids, one after the other, makes our ribosome. Here's the base shape of a ribosome. There's a small subunit responsible for holding mRNA, not necessarily something that you need to know right this second. And then we have our large subunit where the action happens and we actually put the amino acids together. All right, we have two types of endoplasmic reticulum. We have smooth and rough ER, okay? So the big difference is the rough ER, it's actually got ribosomes on it. So it's embedded with all these ribosomes, and it's really fancy. And then here out on the outside of that is the smooth ER, and they do not have ribosomes, okay? So smooth ER functions to synthesize lipids for us. They metabolize carbohydrates, detoxify our cell for us, and then they can also store calcium. Big thing here, they synthesize lipids. Lipids, phospholipid bilayer, so obviously a really important structure for our cell. Okay, the ER that is rough has those protein making ribosomes on them, and so they're going to be the initial place of we need to ship stuff. So one of their main functions is to transport those vesicles and like create those vesicles for us so that they can put that vesicle around a primary structure protein and ship it to the Golgi and actually get it done. Okay? All right. So here's just a base pathway of the endomembrane system. You can see that we'll have information that we've got to get to a ribosome located on the rough ER. We might then put that in a vesicle. We'll put it cis Golgi. It'll modify, trans Golgi, it's done. We'll put it back in a vesicle. We'll ship it to the plasma membrane for exocytosis, where it actually leaves the cell. Okay, so that's just like a base little idea of like how we would move through the cell using the endomembrane system. Okay, so the last couple little organelles that we're going to talk about are the mitochondria and the chloroplast. So both of these are helping us with energy. We call it free energy. Another word for when we talk about energy is we like to talk about the production of ATP, okay, or how ATP is generated, okay? So the mitochondria are responsible for cellular respiration. That metabolic process is what makes ATP by breaking down glucose, okay? We also have the chloroplast, which we find in plants, algae, any of those photosynthetic organisms. And what they're doing is they're actually making glucose for us to then send to the mitochondria and break down for ATP. Okay? So mitochondria and the chloroplast, neither of those are part of the endomembrane system, and here's why. Because they have their own membranes, they have their own proteins, they have their own ribosomes, they have their own DNA. Okay? They are awesome organelles that do everything on their own. So basically, these two organelles, they do their own thing, and we'll find this out in evolution, but they believe that these were like actually originally their own cells, and that's how we got eukaryotes, is through this thing called endosymbiosis, in which a cell basically ate a cell that was a mitochondria or a chloroplast, and then they were able to do stuff. Okay, so here's just a picture of our mitochondria. Another thing that you will notice is that there are folds. We love folds, and we love that there's lots of surface area for us to have reaction sites occurring. So there's lots of folds inside. And then this is just a quick snippet of cell respiration occurring, but we talk about that in a couple units. Okay, another picture of our mitochondria, this time highlighting the fact that it has its own ribosomes, and it has its own DNA inside of the matrix that sends information to that ribosome so that it can actually make its own proteins. And here's our chloroplast, again, responsible for capturing photosynthetic energy, like photons, from light. And again, this is the quick snapshot of photosynthesis, that process, which we like because it's going to make us glucose or sugar down here, which we can then send to the mitochondria to actually break down for us. Okay. Again, also highlighting the fact that this guy in this stuff called the stroma has its own ribosomes studded throughout, because it's its own little functioning thing. And one other dead giveaway is that it's actually got a double membrane. It's got this membrane, and then it's got membranes inside of there, like the thylakoid. Okay, and both the mitochondria and the chloroplast have that double membrane. Well, guys, I hope that helped a lot, and helped you guys get through the cell organelles, and hopefully you guys ask me if you have any questions. The last organelle sorry, totally messed up, is the vacuole. And the vacuole is one that is just, it's a storage. It is a place that we can store waste, we can store nutrients, we can store water, any of that stuff. And these are big in plant cells, and we like them in plant cells. Okay. All right. All right. And just a quick reminder: the cell, it is a unit that is greater than the sum of its parts. Okay, so it's got all of these compartmentalized functioning organelles that work together, but it is needing all of those parts to be working the correct way so that the entire whole can function. Talk to you guys later. Bye.